Immunity, Chapter 7. Integrated Responses to Threats. Immunity is the capacity to resist and combat infection by pathogens such as viruses, bacteria, and fungi. In vertebrates, innate and adaptive immune systems work together to combat infection and injury. Evolution of the body's defenses. Proteins in eukaryotic cell membranes have unique patterns that the body recognizes as self. Cells of multicell eukaryotes have receptors to recognize non-self cues, i.e. PMPs, pathogen-associated molecular pattern, or not-in pathogens, and trigger defense responses. In the picture, macrophage engulfing an anthrax bacteria. 3 lines of defense: physical, innate, and adaptive. First one is physical, chemical, and mechanical barriers. These keep pathogens outside of the body. It is the first line of defense. The second line of defense, you have innate immunity. They are general responses which destroy invaders inside of the body before they become established. Third line of defense is adaptive immunity. Huge populations of white blood cells form to target and remember a specific antigen. Innate immunity. Binding of receptor with the PMP triggers immediate general defense responses that are part of the inborn innate immunity. Complement. It is some proteins that destroy microorganisms or flag them for phagocytosis. It is an innate immune response. As you can see in the picture, the bacteria comes in and then it is activating the antibodies and also complements. So all of these will attack the bacteria, and then some of the complements actually create pores on the bacteria, causing them to leak out and die. Adaptive immunity is a system of defenses that specifically targets billions of different antigens an individual may encounter during its lifetime. Antigen. It is a PMP or other molecule that the body recognizes as non-self that triggers an active immune response. The defenders. White blood cells, leukocytes, are specialized for different tasks, carry out all immune responses. Phagocytes, such as neutrophils, macrophages, and dendritic cells. Secretory cells, such as eosinophils, basophils, and mast cells. Lymphocytes, B and T lymphocytes, and natural killer cells. All white blood cells secrete chemicals, including cell-to-cell signaling molecules, which is cytokines, that coordinate all aspects of immunity. For example, interleukins, interferons, and tumor necrosis factors or TNFs. So as you can see, if there is a stimulus, the cytokine protein cell will activate a cytokine gene, and then cytokine will be created. Cytokine will be absorbed by another cell from a receptor, and the receptor will give a signal to the target cell to activate something. For example, if you are exposed to any peanut butter, if you are exposed to any pollen, cytokine will be released, and then the cell which is in your nasal passages, your nose, will start to create a lot of mucus because they are triggered by the cytokine, and then you will be sneezing. Surface barriers. The normal flora. Billions of microorganisms normally live in human surfaces, including interior tubes and cavities of the digestive and respiratory tracts. A pathogen can cause infection only if it enters the internal environment by penetrating skin or other protective barriers at the body's surfaces. Physical, chemical, and mechanical barriers keep microorganisms outside of the body tissues. Your skin, mucous cilia, lysozyme, especially from your tears, gastric fluids and bile salts in your stomach, normal flora, which is good bacteria, and lastly urination. The skin. The skin acts as a barrier, a kind of sheath made of several layers of cells and their related glands. The skin has normal flora and it is a little acidic. So, in the picture, the skin provides a physical barrier of infection, meaning that infection cannot get in very easily. Other than that, you also have locally produced antibiotics, meaning that your skin cell itself creates peptide antibiotics to kill some microbes. Other than that, you also have intraepithelial lymphocytes, meaning that they are the white blood cells that lives between your skin cells. So they will just patrol around, and then when they find any bacteria, they will kill it. So remember to floss, because in your teeth you have dental plaque. Dental plaque is a thick, sticky biofilm of glycoproteins, bacteria, and their products that contribute to tooth decay and gum disease. 9 of every 10 cardiovascular disease patients have serious periodontal disease. Oral bacteria associated with periodontitis also are found in atherosclerotic plaque, meaning that this plaque will be the collection of bacteria building up a certain layer on your teeth because they want to live there. So sometimes this layer can break off, and then they will enter your bloodstream, and then they will do the same thing in your bloodstream. They will pile up, and then they will block your bloodstream. Innate immune responses. So they will non-specifically eliminate pathogens that invade internal tissue before they become established. You have phagocytes, you have complement, inflammation, and lastly fever. Phagocytes. Phago means eat, so they are cells that eat. Macrophages, they are large phagocytes that patrol interstitial fluid and engulf and digest pathogens. They secrete cytokines when receptors bind to antigen. Cytokines attract more macrophages, neutrophils, and dendritic cells to infection site. Complement. Complement protein become activated when they encounter antigen. Cascading enzyme reactions at concentrate activated complement at infection site. Complement attracts pathogen to infection site and tags pathogen for destruction. Forms attack complexes that puncture bacteria, and lastly help mediate active immunity. So these are the ways that the complement can work in aiding the immunity. First is opsonization. Opsonization is the enhancing phagocytosis of antigens. For example, you have C3b. It has the most important opsonizing activity. Next will be chemotaxis. Chemotaxis will attract macrophages and neutrophils. Cell lysis. They will rupture the membrane of foreign cells. And lastly, agglutination, clustering and binding of pathogens together, which is sticking. Inflammation. Inflammation is a local response to tissue damage characterized by redness, warmth, swelling, and pain, triggered by activated complement and cytokines. Mast cells release histamine, increasing capillary permeability. Phagocytes and plasma protein leak out, attack invaders, form clots, and clean up debris. Fever is a temporary rise in body temperature above the normal 37 degrees Celsius that often occurs in response to infection. Cytokines will stimulate the brain cells to release prostaglandins, which act on the hypothalamus. The fever enhances the immune response by speeding up metabolism and phagocyte activity. But the fever, which is over 40.6 Celsius, can be dangerous. Overview of adaptive immunity. Vertebrate adaptive immunity adapts to different antigens it encounters during its lifetime. Lymphocytes and phagocytes interact to affect 4 defining characteristics: self versus non-self recognition, specificity, diversity, and memory. Self and non-self recognition. Each kind of cell or virus have their unique identity. MHC markers, plasma membrane of your own cell, have sex recognition proteins. TCR or T cell receptors. They are antigen receptors that recognize MHC markers as self, antigens as non-self. So the T cell receptors are receptors on the T cells. They will be the one that will be recognizing whether it is a non-self or self. Self must have an MHC marker. Specificity means that your defenses are tailored to target a specific antigens. Diversity, there are potentially billions of different antigen receptors on T and B cells. Memory, the capacity of the adaptive immune system to remember an antigen. If the same antigen appears again, B and T cells will make a faster and stronger response. First step, the antigen alert. Once a B or T cell recognizes and binds to a specific antigen, it will begin to divide by mitosis. All descendant cells recognize the same antigen. T cells do not recognize antigen unless it is presented by an antigen-presenting cell. Macrophages, B cells, and dendritic cells digest particles and display antigen MHC complexes. Cell types. Effector cells, are differentiated lymphocytes, B and T cells that act as one to fight infection, and memory cells. Memory cells are long-lived B and T cells reserved for future encounters with the same antigen. In this picture, we have the dendritic cell. The dendritic cell will eat the bacteria, and part of the bacteria, which is called antigen, go to the surface of the phagocyte. And these phagocytes present the antigen to a helper T cell. The helper T cell is activated. The two arms of adaptive immunity will be the antibody-mediated immune response and cell-mediated immune response. Antibody-mediated immune response is involving the B cells producing antibodies that bind to specific antigen particles in blood or interstitial fluid. Meaning that you have B cells will create antibodies, as you can see in the picture on top left. Cell-mediated immune response, which is the picture on the right. Cytotoxic T cells and NK cells, natural killer cells, detect and destroy infected or altered body cells. Intercepting and clearing out antigen. After engulfing antigen-bearing particles, intercepting and clearing out antigen. After engulfing antigen-bearing particles, dendritic cells or macrophages migrate to lymph nodes where T cells bind and initiate responses. During an infection, lymph nodes swell due to accumulation of T cells. Antibody-antigen complexes bound by complements are cleared by liver and spleen. Antibodies of other antigen receptors. Antigen receptors on B and T cells have potential to recognize billions of different antigens. Antibody is a Y-shaped antigen receptor or protein made only by B cells that binds only to antigen that prompt its synthesis. Activates complement, facilitates phagocytosis, or neutralizes pathogens or toxins. There are 5 classes of antibodies. The constant region determines 5 classes of antibodies, which you have IgG, IgA, IgM, IgE, and IgD, each with different functions. B cell receptors are membrane-bound IgM or IgD antibodies. Making antigen receptors. Genes that encode antigen receptors occur in several segments on different chromosomes. Different versions are randomly sized together during B or T cell differentiation, producing about 2.5 billion different combinations. T cells mature in thymus, which stimulates production of MHC and T cell receptors. The antibody-mediated immune response. Antibody-mediated immune response starts with antigen, activates the naive B cells and dendritic cells. The naive T cell binds to APC and differentiates into effector and memory helper T cells. Helper T cells bind antigen MHC complexes on activated B cell and secrete cytokines. B cell differentiate into effector B cells, which produce antibodies targeting a specific antigen and memory B cells. Clonal selection and memory cells. Only B cell receptors that bind antigen divide or clone and differentiate into effector and memory B cells. The first effector or primary response produces memory B and T cells, and secondary response is stronger and faster. The cell-mediated response. Cell-mediated immune response starts with dendritic cell ingesting altered body cell, displays antigen MHC complexes, and migrate to lymph node. Naive helper T and cytotoxic T cells bind to APC. Activated helper T cells divide into and differentiate into memory and effector cells. Cytokines signal division of activated cytotoxic T cells. Cytotoxic T cells circulate and touch-kill altered body cells. So, number 1, if the B cell finds an antigen which matches its receptors, it waits until it is activated by T helper cell. Then, the B cell will divide into plasma cells and memory cells. Plasma cells produce antibodies that attach to the current type of invader. The ether cells prefer intruders marked with antibodies and eat slow them. If the same intruder invades again, memory cells will help to activate the immune system to activate much faster. Cytotoxic T cells. Cytotoxic T cells touch-kill cells displaying antigen MHC markers. Perforin and proteases will puncture cells and kill them by apoptosis. Cytokines secreted by helper T cells also simulate natural killer cell division. Unlike cytotoxic T cells, NK cells can kill infected cells that are missing all or part of their MHC markers. Allergy. Allergy is an immune response to a typically harmless substance called an allergen. The first exposure stimulates the production of IgE, which becomes anchored to mast cells and basophils. Later exposure stimulates secretion of histamine and cytokines that initiate inflammation. An anaphylactic shock is severe and potential fatal allergic reaction. This is an allergic reactions overview. First, you have initial contact with an allergen. So your B cells detects it and it will become a plasma cell. And as a plasma cell, it releases IgE antibodies, which is recepted by the mast cell. So mast cell has IgE receptors. So with that, it will actually release a lot of histamine and other chemicals if the allergen comes again. Vaccines. So immunization is an administration of the antigen-bearing vaccine designed to elicit immunity to a specific disease. Vaccine is what's called an active immunization. The preparation containing an antigen that elicits a primary immune response, meaning that you are injected with a weakened virus, a weakened bacteria, a weakened antigen into your body, so that your body will overcome it, so that your body will be protected from that disease forever. Passive immunization. It is an immunization of antibodies which doesn't involve any immune response. This is the story of the smallpox vaccine. Edward Jenner created the first vaccine against smallpox, which has now been eradicated. Noting the common observation that milkmaids were generally immune to smallpox, Jenner postulated that the pus in the blisters that the milkmaid received for cowpox, a disease similar to smallpox, much less viral, protected them from smallpox. So this milkmaid, Sarah Nelms, she is infected with cowpox. So Jane Phipps is inoculated with cowpox pus from Nelms. Phipps falls ill with a mild case of cowpox. The scabs are collected from the smallpox patient. So Phipps was inoculated with the scabs of smallpox, but he is unaffected. He is unaffected from the first infection of the cowpox. Immunity gone wrong. Misdirected or compromised immunity is sometimes the result of mutation or environmental factors. The outcome is often severe or lethal. Autoimmune disorders would be when your lymphocytes and antibodies fail to discriminate between self and non-self. Autoimmune response, it is an immune response, it is misdirected against the person's own tissues, such as rheumatoid arthritis, Graves' disease, and multiple sclerosis. Immunodeficiency. In immunodeficiency, the immune response is insufficient to protect a person from disease. Primary immune deficiencies are present at birth. SCIDs, ADA or adenosine deaminase mutation. Secondary immune deficiency happens from exposure to an outside agent such as virus. For example, AIDS. So primary immunodeficiency, it is a result of mutation. So this girl, Cindy Cutshall, was successfully treated for ADA. ADA is a type of severe combined immunodeficiency SID using gene therapy. So how did she get cured? So all of your cells, all of your immune cells even, are created from your bone marrow. So this gene therapy would use a virus which carries the good genes, meaning that the genes which are not mutated. So they will send this virus to infect her bone marrow, and then the virus will inject the good gene or the gene which are working, not the one that is mutated, into her bone marrow. And then what happens is that the cells in the bone marrow are all kind of naive, all young. So when they're infected with this good gene which is not mutated, they become good cells. They are not mutated anymore. So after many years of therapy, all of her mutated cells are now become the good cells, the healthy cells. So when they are no longer mutated, they have the ability, then her immunity becomes normal again. AIDS, Acquired Immune Deficiency Syndrome. Acquired Immune Deficiency Syndrome is a group of disorders resulting from failure of the immune system due to HIV infection. It includes rare cancers and infections caused by normally harmless microorganisms. Human Immunodeficiency virus, HIV, is a retrovirus that attacks specific cells in the immune system, including helper T cells. So, for the name retrovirus, it is actually an RNA virus. RNA virus, they will actually turn their RNA into DNA so that they can slip into your own DNA. So that's why they are called retrovirus. They are backwards from RNA to DNA. Symptoms of AIDS. Initial stages of infection, no symptoms, but you can still detect the virus. The virus would denote somebody as HIV positive or carriers. After some time, maybe years, it will show symptoms including fever, weight loss, recurring bouts of diarrhea, persistent cough, fatigue, candidiasis or fungus, infection in mouth and pharynx, and swollen lymph nodes. The final symptoms or syndromes of AIDS will be caused by the total failure of the immune system. The affected person will contract pneumonia, which is lung inflammation, Kaposi sarcoma, skin cancer, lymphoma, cancer of lymphatic system. Kaposi sarcoma is never seen in normal people. It only affects individuals with very compromised immune system. As you can see, Kaposi sarcoma are the ones that you see below on the bottom right. Mental disturbances and destruction of the nervous system will occur as a result of infective nerve cells. After 5 years, 20% of carriers develop full-blown AIDS and death within the next 2 years. Mental disturbances and disruptions of the nervous system occur as a result of infected nerve cells. After 5 years, 20% of the carriers will develop full-blown AIDS and death within the next 2 years. Common mode of HIV transmission: unprotected sex, mother to child, shared syringes, blood transfusion. HIV testing: antibodies are found in blood, saliva, or urine. Drug treatment: there is no cure. Protease inhibitors and reverse transcriptase inhibitors can slow its progress. Prevention: vaccines. Experimental vaccines are mostly ineffective or risky. The virus's high mutation rate is an obstacle. Education: the best option of preventing the spread of HIV is to teach people how to avoid being infected. Healthy lifestyle means that you have no unprotected sex, or sex prostitutes, or drug addicts. The end.